For anyone that does not know me, my name is Alyssa Martino. I am a PhD student here at the University of Sydney. For anyone on Twitter and Instagram, you can find me at Myrtle Martino. It has a nice little ring to it. Perry is one of my supervisors, as is Robert Park, who you heard from earlier, and Brian Jones as well. So I just wanted to introduce some of the work today that we have been doing on Melaleuca quinquenervia, so the broadleaf paperbark that you've already heard a little bit about today. So a very quick introduction for anyone that isn't really familiar with the background of myrtle rust in Australia, which I think most of you are. But it was first detected in New South Wales in 2010 and has since spread to all other states and territories aside from South Australia. The climatic conditions here in the East Coast have been really favourable to the pathogen in the past couple of years, and so a lot of anecdotal evidence of there being a lot more infection present, and this was just an image across the road from where I live on some Melaleuca quinquenervia, so that paperbark. And over the past couple of years, these trees have really been knocked back hard by myrtle rust. And what's really interesting is that with the Melaleuca quinquenervia, we do see a variability in the response to the pathogen. So a little bit of what we've seen earlier, also in Michelle's work, that we see some Melaleuca quinquenervia that are highly resistant and respond well to the pathogen, and some of those that are highly susceptible, like what we can see in the image. Something I noticed very early on in my PhD was that we have a lot of, because we have such a broad host range for the pathogen, a lot of the research is spanning a lot of different host species as well. So this work was really to try and build a bit of a model system in a host for myrtle rust research. And there were a few key aims in doing so, but first, just a little bit of a reasoning why we chose Melaleuca quinquenervia. As Bob mentioned earlier, this species is really keystone in our environment, especially here in the East Coast. So we're really having to focus on protecting it to protect our ecosystems as well. It can be grown really easily from seed and propagated, and also we can make cuttings from it as well. It germinates really easily, and we can collect seed all year round as well. So again, as I mentioned, one of the reasons that we really want to work with it, I guess from a genetic perspective as well, is that it shows that variability in response. So it gives us the opportunity to investigate some of those genes that might be conferring resistance. So a few aims of how we're going about this. We have actually just had our manuscript provisionally accepted today for the genome, which is really exciting work. So I'll give a little bit of a background into that. We wanted to annotate the disease resistance genes within the Melaleuca quinquenervia genome as well, and also identify some genes and pathways of resistance. And part of my work was also to see if we can extend some of these ideas into other Melaleuca species as well. So I'll just start with a bit of a summary of the work on the genome. I'm not going to go too deep into the science of it. I know we're all from different backgrounds as well. So if you have any sort of specific questions, I'm more than happy to answer those after as well. So this work was a big collaborative effort between researchers here at USyd, ANU, the Botanic Gardens here in Sydney as well, and UNSW and UWA. So for most genomes, we're probably used to seeing them as one sequence. And we wanted to take a little bit of a different approach with the Melaleuca quinquenervia genome by phasing it. So what I mean by that, if we think of an organism that is diploid, such as Melaleuca quinquenervia, we have two sets of chromosomes, one from the parent A and one from parent B. And by phasing that genome, what it allows us to do is pull those two apart and interrogate the genomes, the sequences from both of those parents. It's really important for our work with the resistance genes, and the reason that that's the case is because some of these are highly variable in plant species and also within a single species as well. So by phasing these two genomes out, it gives us a better idea of that variability that exists within a single species, or a single host, I should say. So again, not going to go into too much detail here, but what part of the work in the genome manuscript is that we are looking for resistance genes, and one of those families of resistance genes is the nucleotide binding leucine-rich repeat genes. So Zenyan, Perry, and some others all contributed a lot to putting together this fine plant NLRs pipeline, which is to identify these resistance genes within the Melaleuca quinquenervia genome, but it can be applied to other genomes also. So we have a high variability in these genes. So without going into too much detail about them, we have different types. So this is determined by those CC and TAR domains. And we have this central NB-ARC domain, which is highly conserved, but that final domain that you can see there, the LRR, is highly variable. And we also have these novel integrated domains. So they're these other domains. I'll go into a little bit more detail on those after as well. But they are additional domains that can be added to these proteins. And we know that there's a lot of variability in how many of these domains are present within particular genes as well. So within the phase genome for Melaleuca quinquenervia, again, I'm not going to spend too much time here, but really all of this is to show that by phasing the genome into those two parental haplotypes, we do see a difference in the number of these genes that are present on both haplotypes, and also the types as well. So those CC and those TAR, they are different. And this has been observed as allelic series in other genomes as well, such as the model Arabidopsis thaliana, or also in flax. So just to highlight here is those integrated domains that I alluded to earlier as well. So we have these domains that act really as decoys. We've heard about effectors today, but they're really targets for the pathogen. And by integrating these into some of these genes, what we actually see is that they can act almost as decoys for the pathogen effectors, which allows the host to respond to the pathogen. So my work has really focused on trying to now identify some of those genes that are conferring resistance to myrtle rust. So I've taken a bit of a different approach in this, in the sense that I am looking at the progeny from the Melaleuca quinquenervia that we use to generate the genome. And there's a few reasons for doing this. Really, the biggest reason was to screen naive hosts. So what I was interested in doing is screening hosts that have never been exposed to the pathogen before and seeing if there's some genetic information that we can capture in the first time that the plants have been exposed to the pathogen. This wasn't a fun task. So I was essentially looking at differential expression between the plants that are showing that hypersensitive response, so that localized cell death, and those that are highly susceptible to the pathogen, so they're showing no response at all. Just a really quick summary into how I've been going about this. So I collected seed from the Melaleuca quinquenervia, so the mature tree that we use to generate the genome. I screened about 220 seedlings initially, just to get an idea of segregation ratios of how many susceptible, how many resistant, to then determine how many I needed to screen to be able to capture enough susceptible and resistant. That's it? Thank you. Okay, 5 minutes, cool. If anyone's met me, you know I talk a lot, so I apologise. So really trying to, so just getting those segregation ratios to get those numbers down, to get enough replicates to be able to work through with a RNA-seq experiment. So from there, I determined that I had to use about 55 seedlings. So I selected 55 seedlings that had never been exposed to myrtle rust before. I collected material prior to inoculation, and I inoculated those 55 seedlings and then collected leaf material at 6, 12, 24, 48 hours post-inoculation. Waited a couple of weeks to score all of those plants, so to get those ones that were susceptible and resistant, and did all of my RNA extraction on 4 resistant and 4 susceptible, and then went on to do the RNA sequencing. So again, this is in fairly early stages still, and because we have a phased genome, I'm still trying to determine the best way to deal with this data. There's a couple of options I can collapse the genomes back down just to work with that one sequence again, or I can be working in a haplotype-specific manner. But what I found was that there was really a large shift in gene expression at 12 hours post-inoculation. So it's a little bit different to what we might have heard today, and this is looking at the host gene expression. But I found that there were about 370 upregulated genes in the resistant versus the susceptible plants, and there was 770 from haplotype B. So between those two haplotypes, there's actually quite a large difference, but I'm still in the stages of determining which ones of those are haplotype specific. And I guess what was really interesting here was just to see that we do see these plants, all of the resistant and the susceptible. So we're just looking at heat maps here of the differentially expressed genes. And we do see this clustering of resistant and susceptible plants, which indicates that they're behaving in a similar manner. What I found most interesting out of all of this, and there's still a little bit of work to do in this space, but as I mentioned, I'm working with plants that are naive to the pathogens. They've never been exposed to it before. And even at 0 hours post-infection, I saw clustering of the susceptible and the resistant plants. So it indicates that, and again, very early on, but it does sort of point towards a possibility of there being a molecular pattern that we might be able to look out for in naive hosts. So even if we have hosts that have never been exposed to the pathogen before, we might have some fingerprints that we can look out for. But the only thing I would say is that I still need to determine how related these plants are. So just getting in the stages of determining that so we can determine whether or not this clustering is really just because they're closely related. So I guess just a bit of the downstream work, which really wraps up what I have been talking about today. So part of my work has actually been screening Western Australian species for their response to myrtle rust as well. So I have screened 13 species to date, and some of these are showing no to low resistance to the pathogen at all, which is really quite concerning. And some of these are probably in regions that are quite isolated. So as an example, the Melaleuca fulgens I didn't see any resistance in that at all, but that's quite widely distributed. But the Melaleuca argentea that we see down the bottom shows a fairly high level of resistance, but that is obviously a susceptible one. And really what I'm hoping to do is try and extend some of the work we have in the Melaleuca quinquenervia and identify some genes that are potentially conferring resistance, and then can we apply those to other Melaleuca species like what I have here. So with all that being said, big thank you to my supervisors, everyone that worked on the genome manuscript as well. Most of you are here in the room today, which is wonderful. The technical staff at UCID and also where the funding has come from as well.
Thanks, Alyssa. And Chris Innes from...
It's a lot of talking. Easy.
Nope. No, I'll...
I'll ask you a question. I've got one too.
Look, this could be a naive question, but when you're monitoring gene expression and your focus is on genes that are upregulated, what about genes that are downregulated? Because they could be equally important if they're negative regulators of resistance, for example.
Yeah, so I looked at both, and the only reason I highlighted the upregulated ones at 12 hours was because there weren't many that were downregulated at that time point. It was really a large shift in upregulation, and so that's why I just chose to focus on that. But it's definitely going both ways, and that's part of the downstream work now is to look at the genes that are most highly upregulated and downregulated as well. Yep. I think Benjamin had a question as well. Hello. No, it might.
Does it go? Oh, there it goes. So you said that you had no to little resistance that you'd found. What did you mean by that? Do you mean just that they all got infected, or are you meaning there was no form of resistance?
Sorry, I realised that was a bit unclear. So no resistance whatsoever. So like when we look at that 1 to 5 scoring system, score 5, all of them. So not displaying any signs of that hypersensitive response at all, just the pathogen living very happily on the plants there. So yeah, still some work to do to determine, what's going on there, but there are some that are very highly susceptible. Just Benjamin, sorry, Jacob.
Nice talk. So, you're screening this quinquenervia population, and I presume it was an open pollinated family.
We believe so, yeah.
Yeah, where you have the mother probably shared. Are you also looking into doing GWAS, another cloning approach of these genes which might confer resistance?
So I think there are other groups already working on that. So my approach was really just with this one sort of host, and the main reason for using seed from the one parent was because I needed to be able to screen the naive host. So I needed to get those segregation ratios to get a bit of an understanding. And I'm hoping to be able to then determine the relatedness, but I think there are other groups that are already working on the GWAS. Yeah.